Hello students, welcome to EPG Pathshala. I am Dr. Shaukat Mir from Hamdard University, Delhi. Today we are going to discuss about a module, Evaluation of Volatile Oils, under the paper Pharmaceutical and Biological Analysis. We are going to learn about following. Number 1, definition and the types of volatile oils and volatiles. We will also look into the chemistry of volatile oils and volatiles. So after discussing the chemistry and the definition of volatile oil, we will then proceed on to various parameters by which we can evaluate oils and their constituents and the products made from them. At the end of this module, we will also try to understand the analysis of individual volatile oils, alcoholic solutions and flavoring extracts. So now let's understand what are these volatile oils. Volatile oils, along with some of those aromatic like benzyl and phenyl propanoids and sulfur containing compounds that are present in many other plants, they make a class of odoriferous compounds used in medicine and perfumery. Volatile oils have a very common misnomer of essential oils associated with it. Actually, this is because of their essence that they have got this misnomer as essential oils. So it is essence because of their good oil smell, they have been called as good smelling oils. All official volatile oils, they are mostly from the vegetable origin. They normally exist in well defined parts of the plants. Sometimes they are present in cavities or ducts. So these cavities and ducts can be schizogenous or lysogenous. So like we understand, they are present in citrus peel, they are present in lemon peel or various like mint, peppermint and various umbelliferous fruits. With the exception of few of those substances, for example the components of bitter almond which are obtained by the hydrolysis of glycosides, most of these volatile oils, they are present in the form of ducts and cavities. They have varied therapeutic as well as other related effects, like they are used as flavoring agents like oil of lemon, they are used as perfuming agent like oil of rose or water or rose water. So besides this, they can act as antiseptics. They can be starting material for the synthesis of other good substances, good smelling substances like ionones, citral that is converted to ionones. They can be used even as preservatives, antispasmodics like most of those ginger, lemon, rosemary, peppermint, caraway and fennel, they are antispasmodic effect, so that there has been a long tradition of their use in our food. Besides, they are also used as aromatherapy chemicals. Now let's understand about the methods of the production of volatile oils. So volatile oils, particularly as we understand, they are volatile in nature, so they can be isolated from the plant matter, from plant matrix or materials by distillation. This distillation can be done by steam. So it can be a simple steam distillation where the plant plant material is put along with water in a distillation flask which is fitted with a condenser, and it is heated. The contents are heated. The steam along with oil, it distills off and it is collected on in the receiver. So where it separates, because the volatile oil and this water, they separate into two immiscible liquids, it can be removed, it can be obtained in the form of an oil. So it can also be done by generating the steam at some different place and passing on that steam onto the material in a utensil or in a flask, so direct contact with water is not in this case, but you generate the steam at one point and then you pass on that, and this that steam takes away the volatile content and it condenses in a receiver. So the another method for the production of these volatile oils is by expression. So expression means you use presses, either mechanical presses or manual presses. So in an industrial setup, they are done by hydraulic presses. So the plant material in subdivisions in smaller form, so it is subjected to expression, high pressure expression. So it takes away the volatile oil which is collected and used after some refinement or cleaning processes. Other methods are very specific and some little bit tedious. One of such methods is enfleurage. You might already know this. Enfleurage is a method where where we isolate the volatile oil based on the property of the solubility of volatile oils in fat. So here on a plate of glass or a metal plate, so molten fat is spread, so it is allowed to stand, and then the sample which contains volatile oil like flower leaves, like rose leaves, they are put on that molten and solid this cooled fat layer. So it takes away that volatile constituents. So after its exhaustion, after 3 hours or even for days, so the new leaves are collected, they are put on this fat layer, then the volatile oil is obtained by solvent extraction. The another method is éculé. So éculé is a method where the samples are ruptured using a needle or projected these utensils or punches. So that, like, it is particularly used in case of lemon oil or sweet lemon oil. So the rind of those samples are punched with with the punches which contain needle shaped projections, and the volatile oils are ruptured. They are obtained after rupturing these cavities. So on the basis of these, we have different categories of volatile substances, either known as concretes and resinoids or absolutes. So absolutes are obtained by solvent extraction. So particularly they are extracted with alcohol, and then alcohol is evaporated, then so that you get the concentrated forms of volatile oils which are known as absolutes. The other modified or advanced methods which are used nowadays is using supercritical gases like carbon dioxide. Low polar, it makes a low polar liquid, so which takes away these volatile oils. And the last of the list is a destructive distillation, which is used in case of for example pinene. Now let's dwell into the composition of volatile oils. We understand that these volatile oils, they are complex mixtures. They are a complex mixture of hydrocarbons and oxygenated substances. So if they are only hydrocarbons, they are known as terpenes, and the oxygenated terpenes are known as terpenoids. So some of the examples of hydrocarbons which are present in volatile oils are like pinene, cymene. So they are present in oil of turpentine, or if the oxygenated terpenes which are known as terpenoids, they are present for in most of those good smelling oils like clove oil. They are present they are present in mint oil, peppermint oil, etc. So only exception in this case is like mostly they are made of either terpenes or terpenoids, while as in case of eugenol, which is present in clove, it is a phenolic. It is rather than terpenoidal, it is phenolic aromatic substance. So the other exceptions are the volatiles from mustard oil and bitter almond oil. The oxygenated compounds are responsible for, or the terpenoids are responsible for good smell and odor of the oils. They are slightly water soluble, for example rose water, orange water, etc. Most volatile oils are terpenoids. Some are aromatic like benzene derivatives, as I told you, like eugenol and camphorol. Some more compounds are aromatic, but still some are aromatic in nature, but still they are terpenoidal in nature, like thymol found in thymine. This slide gives you an idea about the chemical structures of constituents of volatile oils or other volatiles. You can see at the top of it there are these terpenes. Most of them, they are hydrocarbons. They are monoterpenes or sesquiterpenes like alpha-pinene, limonene, sabinine, paracymene, and alpha-terpenine, while a sesquiterpene is beta-caryophyllene. Then oxygenated terpenes, as they are known as terpenoids, they are like linalool, thymol, carvacrol, carvone, or borneol. So you can see that some of them, they are cyclic, aromatic, or even open chain. Besides these terpenes and terpenoids, we have also other volatiles in the form of phenyl propanides like cinnamaldehyde, eugenol, vanillin, and safrole, and still other forms of volatiles are in the form of sulfur containing compounds like allyl isothiocyanate and allicin, as they are present in garlic. So besides this, there are also other sulfur containing volatiles in almond, etc. As has been made clear that volatile oils are products which are generally complex in nature. They consist of volatile principles contained in plants and are more or less modified during the preparation process. The details about the preparation of all official oils has been already discussed. So we know like they are steam distillation, expression, or solvent extraction, and the products of volatile oils and other volatiles are named either as concentrates, concretes, pomades, resinoids, or absolutes. This slide gives an outlook of methods or the parameters for the evaluation of volatile oils and volatiles. You can see that there are some of those parameters based on physical properties, while others are chemical based. Specific gravity, solubility in alcohols, boiling point ranges, refractive index, viscosity, and optical rotation are parameters based on physical properties, while as determination of alcohols, alcohol content, determination of phenols, esters, ethers, ketones, aldehydes, and other volatiles are based on mainly their chemical properties. So besides these determinations, there are also some of those instrumental methods like determinations by GCMS, which are known as GCMS profiles. Official volatile oils and their important constituents. We know the anise is a very important oil obtained from Pimpinella anisum or Elysium verum, which has anethol about 80 to 90%, while as a cinnamon oil, which is obtained from Cinnamomum cassia, it is an aldehyde containing oil, which contains about 80% of cinnamaldehyde. Clove is another example, clove oil, which is obtained from Eugenia carophilus. Eugenol is its main constituent, about 85 to 90% of content is there. Coriander containing linalool, eucalyptus containing cineol, and fennel, which is fennel oil, which is obtained from Funiculum vulgare, contains anethol and fancone. Lemon oil, orange oil are the citrus fruits. These are the oils obtained from citrus fruits, contain mainly the citral or decanol as main ingredients. The last of the list is turpentine. It is actually obtained from Pinus species. It mainly consists of these terpenes like pinene. Let's go to the determination of the contents of volatile oils. The first one is determination of alcohols. The alcohols are determined by acetylation of alcohols followed by hydrolysis of acetates by ethanolic potassium hydroxide and its back titration with N/2 hydrochloric acid. The procedure goes like this. To a roughly measured quantity of oil, add twice the volume of acetic anhydride and a little anhydrous sodium acetate and boil for 2 hours under air reflux. So these acetates are formed. After this, as per this reaction given there, a weight quantity of acetylated oil that has been freed from acid by repeated extraction with brine and water is then boiled under reflux with 0.5 normal ethanolic potassium hydroxide, which hydrolyzes those acetates, as shown in the equation below. The excess of KOH is determined by titration with N/2 hydrochloric acid. In this reaction, in this titration, phenolphthalein is used as an indicator. The total alcohol content is determined using the formula where M represents the molecular weight of the alcohol, X is the milliliter of alcoholic potassium hydroxide absorbed, W is the weight of the acetylated oil taken, and E is the percentage of ester calculated as acetate. The percentage of free alcohols can also be calculated using the same data, using the formula given there, where A is the ester value of the original oil and B is the ester value after acetylation. Molecular weights of borneol, geraniol and linalool, which are the constituents of many of those volatile oils, are 154, while as for menthol and citronellol it is 156 and centanol it is 220. Now let's go for the determination of aldehydes and ketones in volatile oils. So first, aldehydes in volatile oils are determined by hydroxylamine process. It depends upon the interaction of aldehydes with hydroxyammonium chloride with the formation of aldoxime and liberation of an equal amount of HCl, which is determined by titration as per the equation given below. The hydroxyammonium chloride is used in the form of a solution in alcohol and is adjusted before used to a full yellow color of methyl orange by addition of alcoholic potassium hydroxide. This process offers several advantages, that as it is a definite determination of aldehydic or ketonic substance, and also it needs a very small quantity of oil for determination. The determination time is also very short. The procedure for the determination of aldehydes goes like this. A measured volume of oil is taken and it is shaken in a stopper tube with the hydroxyammonium chloride reagent and benzene. The indicator changes its color to red owing to the liberation of HCl. The liquid is then titrated with 0.5 normal alcoholic potassium hydroxide. When free HCl is neutralized, the indicator changes to yellow again, but becomes red again on shaking as the liberated HCl continues. The endpoint is taken slightly past the alkaline endpoint and used as a standard for subsequent more accurate titrations. Aldehyde content is determined by the ml of 0.5 normal alkali adsorbed multiplied 1.008 multiplied 100 divided by the weight of the substance taken. For ketones, the formation of oxime is slow. Thus, tube is heated at 75 degrees Celsius and liberated HCl neutralized at 5 minutes intervals till the final endpoint after 40 minutes. The titration is carried out with 1 normal KOH and correction factor of 0.1514 or 0.1555 are used for calculation for carvone and manthone respectively. This method is official in BP in order to limit the aldehydes in eucalyptus oil. It is also used in the assay of lemon oil USP, orange oil USP and cinnamon oil USP. Let's now understand the determination of cineol in volatile oils. The content of cineol in eucalyptus oil and cajupit oil is carried out by orthocresol method, which depends upon the lowering of the freezing point of a mixture of cineol and orthocresol, which is proportional to the cineol content in the oil. The method is particularly accurate when the cineol content in oil is above 50%. The procedure goes like this. a weighed quantity of orthocresol and oil, which is previously dried with calcium chloride. The oil is previously dried with calcium chloride, is melted in a test tube, which is then fitted through a cork into a bottle, which acts as an air jacket. As the temperature falls and the crystallization begins, the content of the tube are stirred vigorously with a glass wire stirrer. The temperature rises again. The highest point reached, which is read through a thermometer, is taken as the freezing point. The phenol content of the oil is read off directly from the previously constructed graphs in the pharmacopoeias, which are present as appendix. The freezing point should be redetermined if necessary by remelting the mixture until the consecutive readings do not agree. The determination of phenol, in the same way, the usual method for the determination of phenols in volatile oil is by alkali absorption of phenol. The aqueous potassium hydroxide converts phenols such as eugenol in chlor oil to its soluble potassium salts. The volume of unabsorbed oil subtracted from the original volume gives you the volume of the phenol. The procedure goes like, we place 80 ml of 5% aqueous potassium hydroxide solution with 10 ml of clear oil in a flask. We shake the mixture thoroughly for 5 minutes at 5 minutes interval during 30 minutes at room temperature. Raise the unabsorbed portion of the oil into the neck of the flask by gradual addition of more alkaline solution, while rotating and tapping the flask to assess the separation of layers. Allow to stand for 24 hours and read the volume of unabsorbed oil from the lower using the lower meniscus. If the emulsion is formed which does not separate, repeat the experiment after the addition of 2 ml of xylene if needed. Let's now discuss the determination of esters in volatile oils. The procedure goes like this: an accurately weighed quantity of oil or an ester is placed in a flask, and to this add 5 to 10 ml of freshly boiled ethanol, previously neutralized phenolphthalein. Shake well and titrate the free acid with 0.1 normal ethanolic potassium hydroxide using phenolphthalein as indicator. The hydrolysis of ester is then brought about by adding 20 ml of N/2 ethanolic potassium hydroxide and boiling the contents under reflux for 1 hour, frequently rotating the contents. The excess potassium hydroxide is determined by titration with N/2 hydrochloric acid. A blank titration is also performed under the same conditions without oil. Then the ester content is determined using the formula where M is molecular weight of the ester, X is difference in volume of hydrochloric acid consumed in blank and actual test titrations, and W is the weight of the substance taken in grams. The process is applied to determine the percentage of other esters like benzyl benzoate, dimethyl phthalate, ethyl oleate, methyl acetate, etc. that may constitute or that may be present in other volatiles. Another important feature of the analysis of volatile oil is their solubility. It may be defined as the volume of the alcohols of the specified strength and specific gravity needed to produce a clear homogeneous solution of volatile oil at 20 degrees Celsius. In its determination, it is necessary that the alcohol used should be of exactly this required, should be exactly of the specified strength. The solubility of volatile oils is determined in dilute alcohols that are prepared by diluting alcohol 95% by volume with water to 1 liter. The final adjustment of volume are made at same temperature about 20 degrees Celsius as at which alcohol was measured. Dilute alcohols prepared of following dilution and specific gravity ranges are used. So for 90% dilute, 947 ml are diluted to 1 liter, while for 80% dilute, 842 ml are diluted to 1 liter, For 70% dilute, 737 ml are diluted to 1 liter, while for 60% dilute, 632 ml of 95% alcohol is diluted to 1 liter. Their specific gravities are mentioned alongside with them. A solubility value is mentioned in the form of a ratio. For example, a 12 by 19 for lemon oil means soluble in 12 volumes of 90% alcohol. A value of 4 by 9 for ajwain means soluble in 4 volumes of 90%. For almond, soluble in 2 volumes of 70% alcohol, and so on. Another important parameter for the analysis of volatile oil containing drugs preparations is the volatile oil content determination. Many pharmacopoeias prescribe the percentage limits of volatile content of its official drugs. The distillation method is used for determination of volatile oil content from the volatile oil containing drugs and products. Clevenger apparatus is used for the extraction of volatile oils. A weighed amount of drug is placed in a distillation flask with water or a mixture of water and glycerin. The Clevenger apparatus is connected to the distillation flask. To the contents of the flask are heated. On distillation, the oil and water condenses and the volatile oil collects in the graduated receiver after condensing from the condenser. So the receiver has oil and water mixture. So based on the specific gravity of the oil collects either at the base or at the top of the water. So with the help of graduation on the receiver, it is easy to measure the total volume of volatile oil. So on the right hand side, there are two equipments. As I mentioned, that there are oils which are heavier than water or lighter than water. So for these we have two assemblies. A is a Clevenger apparatus for extraction of oil lighter than water, while as B shows Clevenger apparatus for oils heavier than water. So refractive index again is a very important parameter for analysis of volatile oils. It is the ratio of the velocity of light in vacuum to the velocity of the substance. It is determined by refractometer. So it is particularly valuable for purity assessment of volatile oils and fixed oils. It will change if particular oil is mixed with different oils. So it is a very good indicator of determination of adulteration. So refractive index of some of those important volatile oils, like for anise it is 1.533 to 1.56. For cinnamon it is 1.573 to 1.6. Eucalyptus has a refractive index of 1.458 to 1.47. Ginger oil has a refractive index somewhere between 1.488 and 1.494. Clove oil refractive index varies from 1.527 to 1.535, while as lemon oil the refractive index varies from 1.474 to 1.476. Optical rotation is the angle through which the plane of the polarized light is rotated when it passes through a layer of liquid. Many phytoconstituents, including some of the constituents present in oils, have chiral centers, and they have the ability to rotate the plane polarized light coming incident on it. So they can change it either to clockwise direction, which is dextrorotatory, or anti-clockwise direction, which is levo-rotation. If rotation is clockwise, it is designated as positive, otherwise in anti-clockwise it is designated as negative. Such substances are known as optically active substances, compounds, and their biological activity also changes with their optical rotation. The rotation is studied usually at a wavelength of sodium D line at a temperature of 20 degrees Celsius on a layer 1 decimeter thick. So optical rotation being a very important parameter for the analysis of volatile oils. The values of some of those optical rotation are given below for these volatile oils. Like cumin oil, it is a dextrorotatory plus 3 to plus 8. Chenopodium oil, it is a levorotatory which varies, the optical rotation varies from minus 30 to minus 8. Clove oil, it varies from 0 to plus 6 degrees. For lemon oil, it is plus 57 to plus 70. Cinnamon oil varies from 0 to minus 2 degrees. Caraway oil is plus 75 to plus 80, and so on. Specific gravity, as we know, is the ratio between the density of a liquid and a reference substance, which is generally water, at a specified temperature. For water, it is 1 gram per ml at a specified temperature. Specific gravity is the ratio of weight of the oil or melted fat or wax in air at 25 degrees Celsius to that of an equal volume of water at the same temperature. It is applicable to samples in liquid state as that of the volatile oils. So it is determined by any of the two methods. First method, we take a clean, dry, calibrated pycnometer and fill it with recently boiled and cooled water at 25 degrees Celsius. We adjust the temperature of the sample at 20 degrees Celsius and fill it in pycnometer by maintaining the temperature. The ratio of the weight of the test sample and the reference standard gives the specific gravity of the sample. So on the right hand side, we have these specific gravity bottle or the pycnometer, so which are used for determination of the specific gravity. Specific gravity of any liquid can be measured by the other method, which is method 2. In method 2, oscillating transducer can be used. This method for determination of specific gravity depends on the density of the samples. The oscillating transducer density meter is employed for the determination of specific gravity. The density of the sample and water is obtained by using this apparatus, using the formula density of liquid to the density of water at 25 degrees Celsius. Density-based determination of specific gravity of any liquid is measured by an oscillating transducer density meter. Viscosity is another feature which is applicable for liquid samples, although it is more important for fixed oils because they are more viscous, but it is of also importance in case of volatile oils. It is defined as the resistance to flow and shear due to gravity. Viscosity of any liquid is constant at a certain temperature and is an index of its composition, so it is used for the standardization of particular oils. Mostly the viscosity is measured by two temperatures. It can be determined by using rotary viscometer or a capillary tube viscometer. So there are several models available for these viscometers based on rotary or capillary tube viscometers. Now let's go to the specific volatile oils and their parameters that are used for their analysis. For NS oil, the specific gravity is somewhere around 0.978 to 0.992. Its optical rotation is minus 2 to plus 1. Solubility, as have been mentioned earlier, it is soluble in 3 parts of 90% alcohol. So its freezing point is minimum 115 degrees Celsius, melting point minimum 19 degrees Celsius. Cinnamon bark oil, its specific gravity is 1 to 1.035. Its optical rotation varies from 0 to minus 2. Solubility is 3 by 70, which means soluble in 3 parts of 70% alcohol. They contain aldehydes somewhere around 60 to 70%. Going down the list, we find this final oil with a specific gravity of 0.953 to 0.983 with the optical rotation of plus 4 to plus 24, solubility in 5 parts of 90% alcohol, freezing point minimum plus 3%. So the purpose of this table is that all these oils can be standardized, they can be evaluated for their quality using any of these parameters such as specific gravity, optical rotation, or for their content like eugenol, linalool, cineol, etc. So students, let's now summarize what we have learned in this module. So we understand now that volatile oils along with aromatic and sulfur compounds constitute a class of odoriferous, good smelling compounds which have a wide variety of uses in medicine and perfumery. They are light, mobile, highly odorous liquids that can be distilled without decomposition using steam distillation or hydrodistillation and are generally optically active. These are extracted by steam or water distillation as mentioned earlier. The solvent extraction, expression, enfillerage, écoulé and supercritical fluid extractions are the other methods that are used. A number of quantitative tests are used which are based on their physical properties and chemical properties to evaluate the volatile oils and their constituents such as alcohols, aldehydes, ketones, esters, phenols, etc. Certain physical constants of volatile oils are also significant with respect to their ascertaining their quality. So some of those are specific gravity, solubility in alcohol, dilute alcohols, refractive index and optical rotation. These tests and parameters provide a detailed insight of the quality as well as the chemical composition of these substances. Thank you.